Hello my dear students, welcome back. So in the last video I discussed about the centriole and the centrosome. Now today we are going to start the next topic, and it is a very important cell organelle, that is the nucleus. So first thing you should know about the nucleus is about its discoverer, means who discovered the nucleus. So nucleus was first of all discovered by the Robert Brown, that I think all of you know. Clear? Now in this topic we will see what is the content of nucleus, or what is there inside the nucleus. So basically nucleus is a double membrane bound cell organelle. It is a double membrane bound cell organelle. That diagram I will show you. It is a double membrane bound cell organelle. So these two are the membrane. This is the outer membrane. This is the inner membrane. And in between these two membranes a space is there, that is called as perinuclear space. So this space is called as perinuclear space. Perinuclear space. So this is the outer nuclear membrane, inner nuclear membrane, and the space between these two, that is called as perinuclear space. Then what happens at some places, this outer nuclear membrane and the inner nuclear membrane, they fuse together like this, and when they fuse together they will form a pore there, and that pore is called as nuclear pore. This is the nuclear pore. Then what is the function of this nuclear pore? Actually the transport of substances, mainly the proteins, the transport of substances between the nucleus and the cytoplasm, or we can say we can say the locations outside the nucleus. Actually outside the nucleus, what is there? Cytoplasm is there. So the transport of substances between the nucleus and the cytoplasm take place with the help of this nuclear pore. And how this nuclear pore is formed? This nuclear pore is formed by the fusion of the outer nuclear membrane and the inner nuclear membrane. One more thing you should know about the outer nuclear membrane. This outer nuclear membrane, if you remember endoplasmic reticulum that I discussed there, that the outer nuclear membrane is continuous with the endoplasmic reticulum like this. And these endoplasmic reticulum will bear ribosomes over them because they are rough endoplasmic reticulum. So similarly here also, means we can say the outer nuclear membrane is in continuous with the rough endoplasmic reticulum. What about the smooth endoplasmic reticulum? That also I discussed you, that is smooth endoplasmic reticulum, they are the extensions of the cell membrane. But the rough endoplasmic reticulum, they are the extensions of the outer nuclear membrane. Then inside the nucleus, the ground substance is there, and that ground substance is called nucleoplasm. This is called as what? Nucleoplasm. This is called as what? Nucleoplasm. Then inside this nucleoplasm, DNA is there. What is the DNA? But in most of the time, means we can say in the in the resting stage, the DNA is present in the form of chromatin. Actually, this chromatin was first of all observed by the Fleming. Fleming is the name of scientist. So he found that when the nucleus is not dividing, or we can say in the resting stage of the nucleus, inside the nucleus, the DNA is present in the form of thread-like structure. And that thread-like structure is called as chromatin reticulum. It is called as what? Chromatin reticulum, or we can say chromatin fiber. So this is the chromatin reticulum. Chromatin reticulum. chromatin fiber. Clear? Then inside this nucleoplasm, at one place you may find a darkly stained structure. You may find actually one place is there where this chromatin reticulum is very densely packed. Okay, and that structure is called as nucleolus. That structure is called as what? Nucleolus. So basically this nucleolus is not enveloped by any membrane. It is a membraneless structure. Don't think that around this nucleolus any membrane is there. Actually, what is this nucleolus? This is the nucleolus where the chromatin reticulum is densely packed, and as a result of this, it will appear dark inside the nucleus. Clear? That's why it stains very dark, because the DNA, we can see, it is densely packed in this area. Clear? Then what is the function of nucleolus? First I will discuss what is the function of this nucleus. The function of nucleus is that it contains the hereditary information, or we can say it contains the genetic material, or we can say it contains the hereditary information. And that hereditary information is present mainly in the form of DNA. This is the function of the nucleus. But specifically, what is the function of the nucleolus? So this nucleolus is involved in the ribosomal RNA formation, rRNA formation. That rRNA have a role in the protein synthesis because it plays the catalytic role during the protein synthesis. About that you will study in class 12. But here you should know that rRNA plays an important role in the protein synthesis. Clear? So this is all about the nucleus. Okay. So now it's theoretical part in done. Robert Brown was the pioneer scientist. Pioneer scientist means first scientist to discover nucleus. To discover nucleus. Then in interphase nucleus, what is the meaning of interphase? That I will discuss, but here you should know interphase means non-dividing phase. In interphase means we can say non-dividing phase. Interphase, non-dividing phase. In interphase nucleus, the nucleus have an elaborate thread-like nucleoprotein called, that is called what? Chromatin. That is called what? Chromatin. So that you can say chromatin fiber also, or chromatin reticulum. Clear? Nucleus is a double membrane bound cell organelle enclosing a space between both the membrane, and that space is called as perinuclear space. Perinuclear space. Then I told you this outer membrane of the nucleus is in continuous with the rough endoplasmic reticulum. outer nuclear membrane is in continuation with rough endoplasmic reticulum, rough ER in bracket RER. Okay. One more thing I want to ask you, that what is the function of this perinuclear space? This perinuclear space will act as a barrier. Okay, it will act as a barrier between the cytoplasm and the content of nucleus. So that also we have done. Perinuclear space act as a barrier between cytoplasm and content of nucleus. One more point you should know regarding the nuclear pore. Clear? So this nuclear pore is the position where both the nuclear membrane, means outer nuclear membrane and inner nuclear membrane, they fuse together to form a pore. At some places, outer nuclear membrane and inner nuclear membrane fuses together to form a pore called as nuclear pore. Then what is the function of nuclear pore? Nuclear pore, with the help of nuclear pore, the transport of substances take place between the nucleus and the cytoplasm. So through nuclear pore, transport of substances take place between nucleus and cytoplasm. You may ask that which type of substances are transported between the nucleus and cytoplasm. So one thing I want to ask you, where the protein synthesis take place? Usually protein synthesis take place in the ribosomes. Okay, and these ribosomes are present in the cytoplasm. So the rRNA that is formed, okay, not only rRNA, mRNA also, that is called messenger RNA. So that is formed inside the nucleus, that is to be transported into the cytoplasm. So that transport will take place with the help of, means through the nuclear pore. Clear? Now about nucleolus you should know. So what is basically nucleolus? Nucleolus is that place inside the nucleus where the chromatin reticulum is densely packed. Okay, so in the nucleoplasm, what is nucleoplasm? That's cytoplasm of the nucleus. In the nucleoplasm, at some places, I am saying at some places because it is seen that in some cells more than one nucleolus may be there. Usually one is there, but sometime it is seen that more than one nucleolus is present. So in the nucleoplasm, at some places, the chromatin reticulum is densely packed. As a result, they stain very dark and It is called, that is called what? Nucleolus. It is called what? Nucleolus. Then what is the function of nucleolus? Nucleolus are involved in the ribosomal RNA synthesis. Okay, so nucleolus are site of active ribosomal RNA synthesis. Active ribosomal RNA synthesis. Clear? Now one more thing I want to ask you. What is the role of rRNA? So I told you rRNA is involved in the protein synthesis. So rRNA, that is also called as ribosomal RNA. rRNA is involved in protein synthesis. So they are involved in the protein synthesis. Clear? Now a better note. So all these are the components that you will see in the nucleus when the nucleus is not dividing. But what happened? When the cell division will take place, and if you observe the nucleus inside the cell you will not find the nucleus. Rather, you will find the chromosomes. Okay. So in the note I want to give the point that when the cell is dividing, the nucleus is no more, and at the place of nucleus chromosomes are observed, because what happens, the content of the nucleus, they will be converted into a specific structure, and that specific structure is called as what? Chromosome. So during cell division, nucleus disappear from the cell, and chromosomes are formed by the condensation of chromatin reticulum. Chromosomes are formed by the condensation of chromatin reticulum. Means we can say the chromosomes are only visible inside the cell when the cell is dividing. Means if somebody asks you whether in a non-dividing cell you can observe the chromosome or not, obviously not. So basically what is chromosome? We can say chromosome is the condensed form of the chromatin fiber, or we can say chromatin reticulum. Okay. So now our next discussion is regarding the chromosome. So just now I told you that chromosomes are formed only during the cell division. Now one structure of chromosome I want to discuss to you, that you can find the NCERT, very good structure, it is given there. Okay. So on page number 139 you can see that this is the structure of the chromosome. So it is a structure of a well compact, or we can say well condensed chromosome, and here you can see one labeling is there, that is called as kinetochore. What is this kinetochore? Actually this is the point where both these two are the chromatids. This is the one chromatid and this is the another chromatid, and both these chromatids are joined at a point that is called as what? Centromere. And at the centromere you can see on the lateral side of centromere a plate like structure is there, that is called as what? Kinetochore. So this chromosomes are held at their position during the cell division with the help of spindle fiber. So a spindle fiber will be attached to this chromosome at its kinetochore. Okay, means the attachment of the spindle fiber on the chromosome will occur at the kinetochore. And what is kinetochore? Kinetochore is a plate-like structure present on the lateral side of the centromere. So this is the structure of the chromosome. Remember, this structure is only visible when the cell is dividing. Then you may ask when the cell division take place, why these chromosomes are formed. Actually, in this chromosome you can see symmetry is there. On this plane you can see, if you draw a plane here that should pass through the centromere, it will make a symmetry. So what will happen? The DNA, because it is made up of DNA only, or we can say chromatin reticulum, so the DNA will be equally divided into two equal halves. That's why when the cell division take place, so as to facilitate equal distribution of the DNA, this specialized structures are formed, that is called as what? Chromosome. About this I will discuss in the next chapter, that is cell cycle and cell division. Only basic information I am giving here. Okay, so chromosomes are a specific structure, are a specific symmetrical structure which is observed in a cell only during cell division to facilitate equal distribution of DNA. And DNA is our genetic material. Clear? Okay. Now what this means, what is the chemical nature of the DNA? That also should know. Or we can say what is the chemical nature of the chromosomes. So either it is chromosome or DNA, or we can say nucleus. The chemical nature is acidic. So we can say DNA or chromatin or chromosomes are acidic in nature, and can be stained by basic dyes. Dyes are basically color which are used to stain any substance. So DNA, remember, it is stained by the basic dye. Why? Because it is acidic in nature, therefore it will accept the basic stains. Okay. Now, in order to neutralize the acidity of the DNA, one basic protein is present inside the DNA, inside the nucleus, and that basic protein is histone. Okay, so to neutralize acidity of DNA, basic proteins like histone are present in nucleus. So apart from this histone, one more protein is there, that is called as NHC. Okay, non-histone chromosomal complex. That also you will study in class 12. Okay, here only basic things about the chromosome you should know. Clear? Now what happened? On the basis of position of centromere, chromosomes are of 4 types that you have studied in class 10th also. Just as a revision: metacentric, first one is metacentric. In the metacentric chromosome, what happens? Centromere is at center. The second one is submetacentric. So centromere shift towards one side. So what will happen? So that one arm of chromosome is longer and other is shorter. Third one, very simple, acrocentric. In the case of acrocentric, centromere present near the terminal. I am saying near the terminal, not at the terminal, because that happens in the telocentric. In class 10 we also studied that in telocentric chromosome is centromere is at terminal. Centromere is at terminal. So this division is very simple. This division is there on the basis of the position of the centromere. So rest all the studies regarding the chromosomes we will do in the next chapter, that is cell cycle and cell. Only basic thing about the chromosome I discussed here. Detailed study will be there in the cell cycle and cell division. And then one more topic is there regarding the packaging of the DNA into the chromosome, because if you see our DNA inside the nucleus is around 2 meters, but that is packaged in a very tiny nucleus. About that we will study in class 12. So that packaging of the DNA is there in class 12 portion. Okay, so in this chapter this much was required for your knowledge regarding the chromosome. The detailed study will be there in the next chapter, and some part of the packaging is there in class 12. Okay. So by this your chapter is over. This cell, the unit of life, is over. Okay. So hope you understood the chapter, and from the next class I will start the second chapter. That is not, I will not start the biomolecule. First I will finish the cell cycle and cell division. After that I will start the biomolecule. Okay. Thank you.